This is a production of Cornell University.
The title of my talk today is the Malus collection in the U.S. National Plant Germplasm System. Here to show you a publication from the Global Crop Biodiversity Trust 2 years ago. As you can see that in North America, very few actually the crop we use and we eat actually came from North America. So for the longest time, U.S. diplomat actually start collecting germplasm material from around the globe as early as late 1700. And then in 1862, President Lincoln created U.S. Bureau of Agriculture, the current day USDA, and also signed the Morrill-Land University Act, which is very important related to Cornell University. And then move forward 30 years in 1898, 4 major events happened in terms of plant exploration activity. First is the Office of Seed and the Plant Introduction. The current Plant Exchange Office was established by Chao and Swingle at that time. Then that year, the introduction garden for the tropical plant was established in Miami, Florida. That's the first repository in the system. And then also at that year, Neil Hansen was hired as the first plant explorer. And he also introduced the first plant material, PI 00001, Branca brassica oleaceae from Russia at that year, also introduced by Hansen. The plant introductions station later was established at Chico, California in 1904, and also in Savannah, Georgia, and at Glendale, Maryland in 1919. And then in 1946, based on the Research and Marketing Act, 4 plant introduction was established, including the Northeast region, the 89 Project Station at Geneva, New York in July 1948. Here's a picture showing some of the pioneers in the plant exploration activity. Dave Chao and Walter Swingle, who was actually both 26 years old, recent graduate from Kansas State University in major in plant pathology, I believe. And then Neil Hansen as the first plant explorer. Later, he actually became a professor at South Dakota State for the longest time, for over 40 years. One of the most famous plant explorer, Frank Meyer. And also Dr. Charlie Rick at UC Davis. He spent almost the entire career collecting tomato germplasm in the Andes in the South America. And also Harold Olmo, who introduced a lot of grape material in the 1960s from the Mediterranean region into the US. Also Wilson Pomponi spent a lot of time, several decades in Central America, collecting a lot of fruit germplasm material, including avocado. Here's a photo of an interpreter standing in front of a large wild Malus material in the Tianshan region of Chinese Turkestan in 1911. This photo was taken by Frank Meyer. Here's some photo of some of the apple and also crab apple fruit in the market in Xi'an, China in 1914. This photo also taken by Frank Meyer. Currently, there's 20 locations throughout the US that either a laboratory or repository dedicated to the National Plant Germplasm System, including right here in Geneva, New York. At Geneva, New York, there's actually 2 research units. The first is the Plant Genetic Resources Unit, the PGRU. That including the seed crop or the vegetable germplasm collection. My colleague Joanne LeBay talked about the collection last October, also here in the horticultural seminar. And then we have the clonal crop, which is the apple, cold hardy grape, and tart cherry. And also we have a program on the apple rootstock breeding program led by Dr. Gennaro Fazio. And then we have another research unit, is the Grape Genetic Research Unit, GGRU. They're working on the genetics and genome of the grape. Here, just showing you some of the photo from skip one. For the clonal repository, it started actually back in 1974, a discussion at the ASHS, American Society Horticultural Workshop, talking about the need for a fruit and nut crop repository system. And then further discussion in late 1970, and then Bill Cermak, who was a supervisor farm operation, was hired on board in 1983. Bill is actually still working at Geneva. And then in 1984, my predecessor, Phil Forstline, the first curator for the collection, came on board. And the planting of the grape and also apple collection actually began in 1983, and the first planting was in 1986. The tart cherry collection originally was housed at the Davis Repository in California. It was moved to Geneva in 1995 due to better growing environment. Here to show you some of the photo, the view of the collection in the McCarthy Farm South. Just the nursery just started in 1985. Few from 2007 and a few from 2 years ago during the boom time for all the tart cherry collection over there. Here to show you some of the photo of some of the diversity of the apple collection we have over there in Geneva, New York. For the establishment of the Malus collection in the mid-1980s, actually started by donation of a lot of apple material from different programs. And one of the most important is the apple breeding program of Cornell University, actually led by Dr. Roger Way at that time. Here's a photo showing some of the older professor at that time in the horticulture department over there. I believe this is Dr. Roger Way there. And also material from the Purdue, Rutgers, and Illinois apple breeding program, the PRI apple breeding program. Also a lot of material from Washington State University, Agriculture Canada, different arboretum, Glendale Introduction Station, Corvallis Repository. Also the United Kingdom, the British apple collection from the Long Ashton Research Station, and then different material from Australia, China, Germany, Israel, Japan, Netherlands, Switzerland, also Russia. Here's a photo of the PRI breeding program. That's Dr. Julius Jenkins from Purdue University, and he is still very active in the American Society of Horticultural Science. Here, I'd like to show you the distribution of all the wild malus species worldwide. As you can see, majority of the wild malus material is actually located in Asia, especially in China. It is well believed that malus actually origin from southwest China. Then it moved north to central and northern part of the China. And then it continued moving on to the west, follow the Silk Road through history, eventually reach Central Asia, wild species over there, the Malus silvestris, and then eventually reach Europe. For example, Malus orientalis, florentina, and also Malus silvestris, the European crab apple. In North America, there's actually 4 Malus wild species material on the East Coast into Canada. We have Malus coronaria, Malus iowensis in the Midwest, and also Malus angustifolia, the southern crab apple. And then in the Pacific Northwest, we have Malus fusca. The modern day apple, Malus domesticata, is actually a hybrid species. It has most of the material diploid with the 17 set of chromosome. And with their origin actually came from Malus silvestris in the Central Asia, which we'll talk a little bit more, and also from the European crab apple.
Thomas, why are 3 of those species shown in purple?
Those are the Those are actually the Chinese species. Actually, we don't really have them in the collection. So that's, we hope in the future we can get more material from China, also from other area over there. Here is the study we published 3 years ago, looking at the conserved sequence of chloroplast genome of 31 different Malus species and using 460 accession. In general, a lot of Asian species, they all group together, especially those material from China. And then, as you all know, also notice, the Malus fusca from the Pacific Northwest is actually also closely related to a lot of Asian species. Therefore, it is well believed that Malus fusca actually came from Asia through the land bridge when the land was still frozen, and then it reached Alaska, eventually come to British Columbia, Washington, Oregon, and Northern California. And then for the modern day apple, Malus domesticata, and also its progenitor species, they're also actually closely related to the Asian species. And then there's a group of Asian material. They are actually more tropical. For example, the Malus yunnanensis is actually the best example. The morphology and everything of all this material is really different from most of the Asian wild Malus material. So phylogenetically, they kind of group together by themselves. And then we have one very unique material, Malus domari. It's actually located in places like Vietnam, Myanmar, and also into Taiwan. The history of this material is still being debated, but it's possible this material might be actually closer related to Pyrus instead of Malus. So some researchers are working on that. And then we have some Malus material in the Mediterranean region, for example, Fiorentina in northern Italy. But I believe there are some other wild Malus species in Europe, for example, in the highland of Lebanon, and also there's wild species in Sicily. They are really isolated. They kind of maybe have a unique evolution, also domestication history among those isolated species material. And then, of course, we have the North American 3 wild species in the east coast of the North American continent. Here, just showing a chronogram, time frame differentiation between the different wild Malus species. In this particular study, they actually used 2 North American wild species, the Malus alwensis and also Malus angustifolia. You can see this 2 material actually is most ancient compared with all the wild Malus species out there. But they are located in North America, and they separate from all the rest of the Malus material by about 20 to 40 million years. So how these 3 species came about in North America still is one of the major puzzles, at least in my mind. Soon after the establishment of the Malus collection, based on the donation from all the programs, basically from all over the US and also from around the globe, the researcher actually realized there's really the need trying to collect more wild Malus material. And therefore, in 1987, Professor Whedon, who is also working in Cornell University, he started exploration for Malus fusca in Pacific Northwest. And followed by Dr. Elizabeth Dickinson, who was the first PhD student of Dr. Jeff Doyle. And for her PhD at Cornell University, she collected a lot of material, angustifolia, coronaria, also iolensis, and studied their genetic relationship. And then starting in 1989, 93, 95, and 96, Professor Herb Alwinkel, who was a professor at plant pathology department at Cornell University in Geneva, together with my predecessor, Phil Forstein, and also researchers from different institutions, even from different countries, they went over to Central Asia, especially in Kazakhstan, to collect the Malus sylvestre material 4 different times. And then in 1997, they carried out one of the explorations in China, collect 7 different wild Malus species material, and followed by one exploration in Russia for Malus orientalis, and then more in Turkey for Malus orientala too. And then after almost 16-year hiatus in the exploration activity, I restarted exploration activity in 2015 by collecting Malus angustifolia in the southern US. We continue doing so since. This is a survey done once again, by Crop Diversity Trust and also our colleague Gail Volk at the USDA laboratory in Fort Collins, Colorado. They did a survey for all the apple collection around the globe. Here to show you the top 10 apple collection in terms of their number and also their wild Malus species representation in the collection. As you can see at that time, the PGRU collection is the largest in terms of the total number and also in terms of the diversity of the wild Malus species material. Here's the slide I borrowed from Dr. Peter Bradding, who is a national program leader for plant germplasm, also genome in the USDA-ARS. He gave the priority for all the germplasm resources management, all the repository. In general, basically, for all the repository, our #1 priority is the maintenance, also the safeguarding of the material we have in the collection. And for the seed crop and the row crop, that involve a lot of regeneration work, and also safekeeping of the documentation, also data associated with the accession in the collection. All those three activities considered #1 internal priority. And next is actually acquisition. This is either through exploration or exchange of material from other countries or within the US. And the third priority is the distribution of the material to all the stakeholders, that including general public and also researcher, which I will talk a little bit more. And next priority is actually characterization, also evaluation of all the accession in the collection. And the next is actually enhancement. What that means is trying to make crosses, advance the generation, so you can have new trait or disease resistant into the advanced material that can be used by breeder. And then finally is trying to carry out research in support of all the preceding priorities. More specifically for the Malus collection, our #1 priority, of course, the maintenance and the safeguarding of the material. We do that by have a field collection of all the trees, different material, all different variety, and also wild species material. We also use cold freezer, -20 freezer to store the seeds we collected. And also we have using cryopreservation of dormant budwood of the Malus at the USDA National Center for Genetic Resources Preservation Unit at Fort Collins, Colorado. And next is acquisition. We're doing a lot of those activities through exchanges, also exploration of Malus, germplasm in the US, and also from around the globe. And next is distribution. For Malus, we usually distribute between 4,000 to 6,000 samples each year. That including winter dormant budwood, summer budwood, pollen, flower, fruit, seed, leaf, and DNA sample. And then next is characterization. We carry out a lot of different morphology study, also genetic study of all the accession in the collection, and all the information. They are stored at the Germplasm Resources Information Network database, the current day Green Global database. And finally, we carry out a lot of outreach activity, trying to increase the public awareness of the value, also importance of the Malus collection. Here to show you some of the history for the Malus exploration, started by in Central Asia. A lot of Russian scientists actually study the wild Malus in Central Asia as early as recorded back in 1796, all the way in early 1900. The famous plant geneticist, Nikolai Vavilov, actually, he first time explored the wild apple forest in Central Asia in Kazakhstan, first time in 1929, together with one of the plant explorers, Westover from USDA at that time. And then later on, Professor Danilov and other scientists from the Kazakhstan Academy of Science, they explore the wild Malus in the region and also study them for several decades. Here's just a photo showing the first exploration. Vavilov went to Central Asia, the apple forest, the first time in 1929. Here he is, and here is actually Westover, who is a plant explorer from USDA, accompanied him at the same time. Here's some photos showing Vavilov actually came to Ithaca, New York in 1932 to attend the 6th International Congress of Genetics. This is right here, and here is actually a photo of Barbara McClintock right here. And at that time, he came to New York. He also brought with him 2 paintings of wild apple from Kazakhstan and Kyrgyzstan. These 2 paintings actually still housed at the Cornell Library. And this 2 paintings was actually presented to Professor Wellington, who was the director for the experimental station at Geneva at that time. I want to show you, pay attention to this particular painting of this apple. So we'll follow up on that later. Here's a map showing all the exploration in Central Asia by my predecessor and Professor Herb Alwenko and other folks. Basically, most of the exploration actually is in the country of Kazakhstan, but 3 different time locations actually in 3 other former Soviet republic. So altogether, there are 4 different trips in 89, 93, 95, and 96. They have 12 different major sites. And also they collect something they call 44 elite, Malus elite. These are the trees, when they're in the field, they look wonderful. Maybe they show resistance to diseases or drought or some other unique fruit quality. Therefore, they also collect the budwood of those 44 material, bring it back to the US through the quarantine system. But majority of the material they collected actually the open pollinated seeds. Altogether, they collect 894 seed accession from Malus in that 4 different trips. And after altogether, they collect over 130,000 seeds. After return to the US, over 40,000 of those seeds actually was sent out to all the apple researchers in the US, also around the globe at that time. Up till now, over 39,000 of those seeds from the original exploration still stored in the freezer at full column, and over 50,000 of those seeds actually stored at the freezer in Geneva. Also, in the last 10 years, we've been collecting open pollinated seeds, over 200,000 of those OP seeds from Malus seeding block. We call those a bounty of desire seed because, thanks to Michael Pollan's book, a lot of people asking for the material. So when people send us a request, we'll send a package of 25 open pollinated seed of Malus with the instruction how to stratify and germinate the seed. So you can have your own wild Malus seed from Central Asia. Here to show you quickly some of the photo. And as mentioned earlier about that particular painting from their exploration in 1980 and 90, one particular apple really similar to the drawing all the way by presented by Vavilov to Professor Wellington at that time. Some of the photo exploration in China in 1997, exploration in Turkey, the route they take all over Turkey at that time. And then in 2015, I restarted exploration activity at Geneva, New York, with funding, once again, funded by Plant Exchange Office, National Germplasm Resource Laboratory. It was a 16-day trip, almost 5,000-mile driving from New York down to Arkansas, Mississippi, Louisiana, and Alabama, and came back. I was able to collect over 7,000 open pollinated seeds from 38 different trees, also some of the leaf sample at that time. Here's Mark Alfred, who is a professor at University of Southern Mississippi, who was actually also a graduate from Cornell University. He was very helpful for this trip. And then follow-up exploration, once again, in Arkansas, Mississippi, Louisiana in 2016. Almost 5,000 open pollinated seeds from 18 different trees, and also a lot of more budwood material at that time. And last year, together with the Julian group, we actually went to Pennsylvania to collect the Malus coronaria material. We're able to collect over 12,000 open pollinated seeds from 29 different trees. Also, 11 different locations. Actually, we find there's a lot more Malus coronaria trees in Pennsylvania. So a lot more sample could be collected maybe in the future. A week later, I went on to another trip by myself to collect Malus coronaria and also Malus ioensis in Michigan, Illinois, Iowa, and also Missouri. One of the purposes of this trip actually was following the footsteps of Elizabeth Dickinson 30 years beforehand. And I visited 22 locations that Elizabeth actually collected back in the late 1980s. Unfortunately, 20 out of 22 sites that he collected, sometime her description was saying there's over 100 trees in particular location. All those 22 20 sites, all the wild malus trees are all gone due to a lot of development in the Midwest. So some of the lessons I've learned from the malus exploration so far in the US. The first thing is trying to understand the role of the target species play in the ecological setting. It's actually very important to understand its location, the age of the environment. It's also affecting the growth habit of the tree, its morphology, and also how reliable is the herbarium sample that we base on for collection. A lot of older herbarium sample, by knowing the possible location and environment this species could be, then actually you can eliminate some of those locations. Most likely the tree sample will not be there anymore. There's actually a lot of sites they were lost, especially during the last 30 years due to urban development. Especially seems like that's the case in the Midwest. And also in the South, actually due to a clear cutting logging practice, sometimes they will come in, chop off entire block of trees, including some of the freestanding wild mouse material. Also seems climate change could be a problem. Hurricane in the South, sometimes flooding. Actually also down there, some of the tree, they've been around for maybe 10 years. I've seen that in 2015. But on 2016, when I returned to the same site, almost trees along a mile track, actually on both sides, are all flooded. All the trees just got killed in that year. And also a lot of land management decisions, especially a lot of wild malus species material, they are very thorny. So in a lot of the park location, people actually would cut a tree off because the tree probably are too thorny because of that reason. But I believe there's still a lot of wild malus material still exists, especially in public forests, state park, nature reserve, and even some of them actually on private land. So, but they're getting more difficult to find. But I think we should try to spend effort trying to get more before they completely disappear. Then talk about why we're trying to collect, preserve the wild germplasm material, especially the wild malus material on a global scale. A lot of material, also their natural habitat are lost in the last decade or so, and also last half century, due to a lot of agricultural development changes, and also the changes. Also a lot of urban development, also a lot of human activity, especially in some less developed countries, such as firewood gathering. It's really a problem. People just chop off all the wild malus in that case. Also a lot of animal grazing, environment degradation, climate change could be an issue, also land management decision. And also in some locations, especially like in Europe, that's the case for European crabapple, Malus sylvestris. The gene flow and also pollen contamination from the cultivated species really become a serious issue. In some cases, the study showed that 60% of the wild, the open pollinated seed you collect from 60% of population, actually all contaminated by Malus domesticated pollen. So that's a serious issue in Europe. Sometimes due to new pests and disease and also new invasive species. So a lot of problem. Also climate change that's been documented in Central Asia, in Kazakhstan, for example. In the last almost 100 years, temperature has risen, both in the winter time, also during the springtime. There's some study from China, for example, in the 50-year period, 80% of the wild mollusk material in Xinjiang, China were all lost due to a variety of the reasons. One of the major reasons, surprisingly, over there is actually overharvest of wild fruit. The local resident, actually, they like to collecting the material. So in the last 2 years, the researcher actually trying to collect more material, but they couldn't because before they showed up, all the fruit were gone, collected by local resident over there. So that's a serious problem. And also there's a study in 2014 showing that firewood gathering is especially serious issue in Central Asia. A lot of wild mollusk material, also a lot of fruit tree material, simply just being lost. So they can be completely gone in the next 50 to 150 years. There's some target of future Malus exploration. I'm going to collect more cornaria in Ohio this year. My colleague Gail Volk, she's trying to carry out exploration for Malus damari in northeast Vietnam this fall. Next year, maybe more Angusfolia in the southern US, and also Malus coronaria in West Virginia in 2 years. And then there's a lot of material in different places. That's our wish list. So we're trying to collect, hopefully in the next 5 to 10 years. Besides go collect the plant germplasm material, either in the US or also around the globe, we also do a lot of importation of the Malus germplasm material from other countries into the US. All this material we bought in has to go through the USDA-ARS quarantine system. It has to be cleaned up over there, then we'll release to us. Every year we receive between 10 to 20 accession from the ARS, sometimes more. In recent year, for example, the Malus silvestris elite from Central Asia was imported. And also a lot of cider apple from northern Spain was introduced actually by Professor Ian Merwin before he retired. 2 years ago, a lot of cider from England was introduced by Greg over here. And then a lot of different variety and rootstock material from a variety of different countries in the last 5 years. And also, all this material takes several years to go through the quarantine system. One of the most extreme example are 2 of the Malus elite material was collected in 1996 exploration. One of them was just released 1 month ago. We got notice from APHIS saying after 22 years, the material is finally released. There's one more actually still in quarantine, maybe next year. So we'll wait and see what happens. So altogether in the last 5 years, we introduced over 162 clones. And also we collect over adding 306 seed accession to the Malus collection in the last 5 years. We also have been selecting a lot of wild seeding material and add to the permanent collection. We add over 300 material accession to the permanent collection in the last year. So altogether, almost 800 different new accession were added to the collection in the last 5 years. Here is just show you why the APHIS quarantine system takes so long. One of the major reasons is it has to go through biological indexing. If there's any disease being found, then they have to go through a tissue culture process, then go through a thermotherapy, and then go through shoot tip grafting, micrografting of the material, and then being retested again before they can provisionally release to us. Then we're based on field observation for additional 2 to 3 years before they can fully release. So normally this process takes about at least 5 years. But in the extreme case, we have a material being in the quarantine for 23 years now. So last year, there's an interesting development. The Global Crop Diversity Trust, together with the Millennium Seed Bank at the Royal Botanic Garden in Kew, England, they have an ongoing project. They're collecting 29 crop wild relatives in 24 different countries, including the wild Malus. This project was funded by government of Norway. And for the Malus collection, they decide all the seed they collect, actually 1/3 of all the seeds will stay at the country of origin. The other 1/3 of the seed will be stored at the seed bank in Kew Garden. And after quite an intense discussion for almost 2 weeks, they made a decision. The remaining 1/3 of all the wild Malus seed collected will come to Geneva for evaluation, also characterization. This including wild Malus from 7 different countries. Some of the material from this country will never have that in our collection, so it's very interesting and exciting, hopefully to get the hands on this material from 7 different countries. Next, I want to tell you some of the story we get involved in the last 5 years. For example, in 2016, 2 years ago, we propagated a whole bunch of 28 crab apple from belong to different wild Malus species. Some of them are hybrid species from the U.S. National Arboretum. They have a new director over there, and he made a decision. They're going to remove their entire Malus collection. So some of the material we're able to rescue and come to Geneva before they were completely removed. And also, back in 2017, last year, one of the material is Malus halinana from China. It's actually located at USDA Chico Station since 1914. It was originally collected by Frank Meyer. And once again, also in Sanxi area, he collected material in 1914, January, and then sent to Chico Station in March. The material actually has been there for the last almost 100 years, okay? Somehow, a colleague from the National Plant Germplasm System, Gary Canard, and also Karen Willen was able to go to Chico Station, identify the material, and then the bubble was sent to us for preservation. It was very interesting. This is actually multi-petal, layer petal of crab apple material, wild apple material. So it will be interesting to see how it looks like right here in Geneva, New York. And also Karen Willem and also Gary Canard, they also went to former USDA station, introduction field station in Cheyenne, Wyoming. They found a whole bunch of Malus material has been around since about 1920 and 1930. So they're able to rescue about 11 material, and we're going to collect the bubble and send to us for propagation. Some of them, several red crab apple material from Canada. We call that Lake Series crab apple. And also 3 different wild Malus material, either originally came from China, collected by Dorset in 1927, or from Sichuan, China, and also 1 material from Europe. So those material rescue will come to Geneva, New York. Next, I want to talk a little bit about the maintenance of the Malus collection. Basically, all the variety, we have trees in the field, and also we have a core collection. For Malus collection, we have restored the seed in -20 freezer. We also have the dormant bubble in the cryopreservation. Here is showing some photo from the center in Fort Collins, Colorado. We test the viability by budding. And then for our routine maintenance of safeguarding of the material, we do a lot of weeding, spraying, nursery operation, also pruning during the winter time. Here just showing the list of all the collection we have. We have over basically 7,000 accession right now in the total collection. All the permanent material have a tag showing all the information. A photo of the collection. Here's my predecessor, field force line, and Dr. Herb Alwenko. In 2013, during the bloom time. Here just showing some of the data from our distribution record in the last 8 years, basically between 2010 and 2017. We distribute basically, this is the number of requests we got in the last 3 years. You can see in the last 3 years, we really have a high number of requests. Actually, 2016 was a record number of requests. But it was the total number of sample was not a record, but we believe that 2018 might be a record. We're doing a lot of characterization work. All the information about the material actually stored in the Green Global database. We also do a lot of study about the chemical composition, the disease and information. All that is all stored in the Green Global database. We also, in the last 20 years, did a tremendous amount of work in study the wild Malus germplasm material. Basically, a lot of disease-related work, and also study of the material, different trait. This is one particular materials actually able to discover a new, brand new, highly resistant material against blue mold in Malus. Here just showing the inoculation, 7 days after inoculation in the resistant material versus the susceptible material. It's a significant contrast between the reaction after inoculation with blue mold. And two major QTL control this trait will map in chromosome number 3 and chromosome number 10. And then we do a lot of other study, different trait with a lot of different cooperator. We'll also provide a lot of material for different researchers everywhere. One of the best examples is actually the publication by Velasco in 2010, the genome of the domestic apple. Actually, in that study, they used 74 accessions. Actually, 46 of those accessions actually all came from Geneva collection. So, in the last 2 years alone, there's about 18 publications. We are directly involved with 7 of those, using our germplasm material to carry out the research. We did the GBS work since 2012. Did a GWAS analysis using the GBS data. Also, we did an extensive resequencing study with Dr. Liang Chen and Dr. Fei of the Thompson's Research Institute right here in Geneva, in New York, in Ithaca actually. Study the history of domestication of a modern day apple. Here's showing the diagrams, actually 2-step process for food size increase. One happened before domestication process, one actually happened during the domestication process in Central Asia. A lot of work was together with Greg in the last few years on searching for the hard cider for New York, which has become one of the fastest growing sector of the industry for alcoholic beverage industry in the US. Greg has studied the Malus collection we have, looking for the best material. Also, the Spanish cider material, also right now in the collection. We're trying to look into some of the wild mouse material. This is a photo of actually a cider made from a red flash material, almost look like a red wine. Then we are currently actually supporting 19 different ongoing research. A lot of them long-time cooperation, for example, with Dr. Susan Brown, the apple breeding program at Cornell University, and then also the apple rootstock breeding program, and a lot of different work in the last couple of years. People from Michigan State and Purdue University, Dr. Kinan Su, right there in Geneva, doing a lot of food quality work and also tree architectural traits analysis. We're doing a lot of sequencing work with different researchers from different institutions, from Harvard University, from Jiangyin Institution in England, some folks from Australia, also some disease work, and also insect monitor work, and also with Greg on the cider work, and also with Dr. Liang Chen on the beta raw evaluation in the last 2 years with Ben over there. Also, right now with Dr. Awaz Khan, who's a new faculty, relatively new faculty at Geneva and plant pathology, working on different disease-related issue. And also, we're supporting a lot of cider variety testing right now because cider is getting so hot, a lot of people are interested. So we're actually supporting material for study in actually places like Arizona, Georgia, California, Maine, Michigan, Montana, New Mexico, New York, of course, Washington State, and also West Virginia. We study a lot of those material. So then we have some unique requests in the last couple of years, especially I think this request from the Smithsonian, trying to get the bark tissue from a native wild species from Japan to study the yellow dye used in the traditional Japanese painting. That's also very unique. We're doing a lot of outreach. A lot of people visit our collection. Hobart William Smith College classes, several classes every year. And also CBS Sunday morning, Moraka, last year, October, visit the collection. Cornell plant breeding courses, 40, 60, actually come to the collection every year in September. And also a lot of researchers from different countries, from China, from Brazil, and from many other countries. Also the grower, extension personnel come to the collection all the time. In the last 5 years, basically over 1,500 visitors actually come to the collection. 79 different groups and classes. So it's very interesting. Finally, I want to thank a lot of people involved in the project, and it's a very long list. Here, just showing some of the photo of some of the folks we worked together on the project. And any question? Thank you. Sorry, I have to rush through the latter half of the talk. So, oh, Liang, please.
Hey, Thomas, thank you for the presentation. Really interesting. I have two questions. One is technical. The other is more of a looking into the future type of question. The first one is, in that phylogenetic tree, you showed that there is a sorbus outgroup there. How many members are in that group, or how many accessions in that group?
I don't quite remember. I have to go back to look. Sorry about that. Liang was asking in the chloroplast DNA sequence study of the Malus relationship. We have a sorbus outgroup, so he's asking how many accessions in our group. I don't quite remember, so I have to go back to look. Sorry.
So for the second question,
yes.
Looking to the future, how do you think the PGRU should adapt to the research needs in the post-genomics era? Because currently PGRU largely keeps 1 or 2 trees per accession. For sequencing work, that's absolutely fine. But for any other type of, let's say, metabolomics or any physiology studies, or I think 1 or 2 reps are just, don't provide good information. So do you think this situation is going to get improved in the future, in the next few years?
The question Liang just asked is how we're going to adopt maybe in the future regarding currently that most of our collection in the field, we only have a single tree representation. So that's probably enough for genetic related study, but that is probably not enough for additional kind of physiology or other kind of studies. So we have any idea how we're going to move forward in the future regarding this? That's actually quite an extensive discussion on a continuing basis for ourselves, and actually also for the entire clonal germplasm repository system. We're facing a limitation on the resources and also on the land, also on the personnel. So it's very difficult trying to have more than 1 copy of the trees. Most of the repository actually have more than 1 copy. The reason we go with a single tree is because we do have a very good cryopreservation backup system right now. About 95% of all the permanent collection we have in the collection, actually they are back up in the cryopreservation at Fort Collins, Colorado. So that's the reason we go with a single tree representation. I think moving forward, there's discussion, trying to get some funding support, maybe trying to have, create maybe a new core selection with better representation of the entire diversity, and also including some of the more important variety from recent in the last 30 years. The core right now we have is almost 30 years old. So a lot of information, different selection could be including maybe this new core. So if that's the direction we'll go with, then hopefully we can maybe have duplicate of the new core, maybe 2 or 3 of those duplication, either planted in Geneva, additional site in Geneva, or maybe trying to work with other university, such as the Washington State University, and also maybe with Kernersville, West Virginia. They also have land over there, so we have a copy of the new core over there. So if we can do that, then we can probably at least minimize some of this single representation issue for the future.
Thank you.
So.
Nice presentation. I know that a lot of people are really obsessed with apples, and I'm sad to see that so much of the genetic resources are disappearing. And so to tap into people's obsession over apples, have you considered engaging the public in some of these what do you call it, citizen science, or some type of project where they could report to you what they think is an ancient tree on their property or along railroad, like old, like railroad, what do you call them, connecting east to west or east to south. Because I've heard about this in Washington, that there was a nursery where it's all a bunch of ancient tree varieties, and they primarily collected it from abandoned farms and railroad sites. And so I wondered if you could engage them, people around the United States to give you the seeds or report to you where these are located.
Right. Actually, you're correct. There's a lot of public interest in apple, specifically on something we call the heirloom apple variety in North America. Actually, I went to one of the first heirloom apple meeting last year in Maine. So there's actually a different group, especially a very active group in Maine, in Colorado, in Washington. There's actually a story just came out from Washington State about discovery of a new heirloom apple variety. And also, we're working with a history professor actually in Georgia. He's working on 15 heirloom apple variety. He kind of rediscovered over there. So yes, we got constant requests actually by the heirloom folks, but it's very hard because we have limited space. So a lot of private actually institutions stepping in this regard. For example, the Sea Saver Exchange in Iowa. Currently, their collection for heirloom apple is only about 400 variety. They actually plan to increase their collection of heirloom variety up to 1,500. Then there's a Southern Heritage Apple Orchard in North Carolina. Their collection currently have about 2,000 heirloom apple variety. And the group in Maine, Maine Heritage Foundation maybe, they currently have a collection of about 300 to 400 heirloom apple variety. And then there's more this going on in Colorado and Washington State. And also, actually, there's some interest. I got phone call and contact from some of the folks from New York, actually just 2 weeks ago, from western part of New York, and also from other part of New York, people asking a lot of this question. We're trying to help, we're trying to engage, but we're also limited what we could do. So that's a very challenge. But we'll help them. For example, if they want to engage in genetic study, so they need a standard variety, usually they will come to us. We'll provide a sample for them to do comparison genetic study, for example. We actually do that all the time. And, also, the Heritage Apple Group, actually, they are considering trying to come to Geneva for their annual meeting, actually, this year. They want me to chair the organization committee, so we'll see how that goes. It's going to be a lot of work.
I want to thank
Thanks.
Professor Chao for a fascinating presentation. Thank you.
Thank you. Thank you for coming.
This has been a production of Cornell University, on the web at cornell.edu.